And I'm going to go full screen. Now, I am going to share my screen and get down to business. So what we are talking about today is glycolysis. The slide deck that I'm using today is titled Week 8, Day 1, Glycolysis plus Enzymes Day 3. And the reason for that is because, one, this is where we are in the class. But the reason that this is day 3 of enzymes is because what we're doing is we're integrating a discussion of sugar metabolism, this process known as glycolysis, and an introduction to enzymes, um, and specifically the categorization of different enzymes. Now, with that in mind, I'm going to go ahead and get started. So, glycolysis. Could everyone give me a number yeah, a number in the chat if you have ever heard of glycolysis, a letter in the chat if you have never heard of glycolysis. Okay. Typically it's if you're unfamiliar with glycolysis, it's a pathway that, people will mention in like an introductory biology class. And it's kind of addressed and painted with a broad brush. Oh yeah, glycolysis. That's what that is a metabolic pathway. If you elect to go on to take a metabolism class after this, which I highly recommend, taking Chem 4385, you will learn about not only glycolysis, but other metabolic pathways and how all of these different molecules that we talk about, how they're utilized. So glycolysis, the purpose of it is to take this 6 carbon molecule right here, this 6 carbon molecule known as glucose, and split it into a pair of 3 carbon molecules. So in terms of, I always like to talk about glycolysis in terms of kind of accounting. So we start out with 6 carbons in one molecule, and then we ultimately, we make some modifications to it and we end up with 2 3 carbon molecules. Now, what I'd like you to recognize about this is, well, in order to break a 6 carbon molecule into 2 3 carbon molecules, it's pretty reasonable to say that you're going to have to break a carbon-carbon bond. Now, a carbon-carbon bond is a covalent bond, so it's a high energy bond. Now, that's what I want you to think about. But if you look at this molecule right here, one of the things that I want you to recognize is we've got hydroxide group, hydroxide group, hydroxide group, hydroxide group, hydroxide groups throughout that 6 carbon molecule. And then if you look at your pyruvate down at the bottom, carbonyl oxygen, carbonyl oxygen. Okay. Okay, we got a pair of carboxylic acids. So long story short, to go from an OH to a carbonyl oxygen, you're doing an oxidation reduction reaction. So we're going to be doing lots of oxidation reduction reactions throughout all of this. Now, we're going to get into that a little bit more in just a moment, but what I want you to know about glycolysis is that we are going to invest adenosine triphosphate, also known as ATP. We're going to invest inorganic phosphate, or PI, ADP, and NAD+. We will recover ATP and NADH. Okay, so think about this as you input a couple of things, you output different things. Glycolysis shown here is a series of 10 steps. We're going to go through each one of those steps, and I'm going to give you some ideas on how to learn this pathway with kind of some ease. Now, just the other day, I made a playlist on my YouTube channel of, let me get it. I'm going to copy this link. Of the 10 steps of glycolysis kind of simplified. I'm going to add it to the chat right now. So that playlist right there just it has one overview video and then 10 steps or 10 videos that go through the different enzymes, the different reactions in glycolysis. So that's, kind of summarized. And they're all short, so it's basically, maybe 10 minutes worth of content. So that's just something if you wanted to kind of watch it as a quicker. Okay, so our learning objectives for glycolysis, I want you to identify 6 different classes of enzymes. Oh, I haven't talked about classes of enzymes. That's what we're going to do next. And then the rest of this is all applied to glycolysis. So I want you to know the classes of enzymes. Then I want you to know the overall purpose of glycolysis. Purpose of glycolysis. I want you to know and be able to outline the phases of glycolysis. I want you to summarize the net reaction, the overall reaction of glycolysis, the enzymes of it, the irreversible steps. I want you to know the difference between hexokinase and glucokinase. I want you to know the net yield of ATP glycolysis and explain the role of NAD plus in glycolysis. I'll try to revisit each one of these points periodically, so that means I'll toggle back and forth between this slide and future slides. Now, is there anyone who's preparing to take the MCAT? If you are, add a letter to the chat. Okay, cool. So if you're preparing for the MCAT, first and foremost, I wish you best of luck. That's an intense exam. I want you to do well on it. And I'm hopeful that what you learn in this class and in this particular lecture will be beneficial to you. Now, if you're preparing for the MCAT, one of the things that you're talking about whenever you discuss enzymes is you talk about classes of enzymes. And these classes, you could describe them as types of reactions. If you think back to, say, Gen Chem 1, I remember taking Gen Chem 1, teaching Gen Chem 1, and you learned about a couple of different reactions. You learned about a synthesis reaction. You learn about a decomposition and a combustion reaction. These are different types of reactions. Now, enzymes do the same sort of thing. The difference is that we kind of break it down a little bit more than that. Now, if you're preparing for the MCAT, chances are the materials that you're looking at don't describe the 6 major classes of enzymes, but instead the 7 major classes of enzymes. So there's all these, which I'm going to highlight or circle these major classes. So these 6, and then there's a 7th one that are your transferases. Now, with that to say, what I want you to know about this is I want you to know this number right here and the type of reaction that it catalyzes. So whenever I think about these, is anyone familiar with like an IP address for their computer? Give me a Y or an N in the chat if you're familiar with an IP address. Perfect. So if you're familiar with an IP address, chances are you might look it up on your computer and it might be something like 10.2.17.164. And I am not a IT person by any stretch of the imagination. I know IP stands for Internet Protocol. But to my knowledge, what these different numbers indicate is they kind of narrow it down. For instance, this set of 4 numbers is going to be linked to your specific device. And that might be a router or something like that. And if there's anyone who is an IT expert and I say something that's incorrect, please, I apologize. I'm doing my best with my dysfunctional IT knowledge. So this set of numbers will be like your home computer. So I'll just say my laptop. Whenever laptop at home. Obviously, when I go somewhere else, it changes. But this is my laptop, and let's say that this is the IP that I always get when I'm at home. These 3 numbers will be something like, oh, well, if I have those 3 numbers leading, that's going to be, well, for my apartment complex. And then these 2 numbers would be something like my internet connection or my ISP, my internet service provider, ISP, and I'm going to say regional. My regional ISP, and then this first number is going to narrow it down to say like in the USA or maybe Texas or something like that. So my point is that this collection of four numbers, you could start with that first digit and say, okay, well, I'm looking I'm looking at these numbers and I can say if 10 is the first number, that is a computer that is in Texas. Okay. 10.2. Okay, well, that is a computer in Texas in the southwestern portion of the state. Okay, 10.2.17. Okay, well, that's a computer that's in Texas in the southwest portion of the state at Bobcat Village Apartments. So you kind of narrow it down. Now that's where I'm going to stop my comparison to an IP address, and instead I'm going to erase all this. And what I'm going to do is instead go back to these numbers. So the leading numbers. So if you have a number of one for your to describe an enzyme, that enzyme is catalyzing an oxidoreductase reaction. So that means that there's electrons that are being pushed around. You're doing an oxidation reaction or a reduction reaction. If the lead number for an enzyme is 2, it's a transferase, so you're moving a group. 3 is a hydrolase, so you're using water to break a bond. 4, you are adding a group and you are using a lyase. 5 is an isomerase, so you're rearranging the atoms. 6 is a ligase where you're forming a bond with ATP being or harnessing the energy of ATP. And then number 7 is a transferase. So something's being transported from one side of the membrane to another. Now, like I said, I want you to know the 6 major classes. If you're preparing for the MCAT, you'll need to know the 7th class, which is your transferases. The reason that I'm not integrating that as a part of our instruction is because of the fact that these transferases, it's a fairly new grouping. I think they came about in like 2016 or 2018. And what they related to and what a transferase does is actually a series of reactions. So a transferase might have elements of a ligase, isomerase, and a lyase present. But the net reaction was to move something across a membrane. So what I'm going to show you next is some examples of these reactions. What I want you guys to know is I want you to associate 1 oxidoreductase, 2 transferase, 3 hydrolase, 4 lyase, 5 isomerase, and 6 ligases. Now, here are your examples. So I'm going to show you this enzyme right here. I'm going to remove that. This is an enzyme known as lactate dehydrogenase, or let me rephrase that. The enzyme lactate dehydrogenase is an enzyme that when it's provided with lactate and NAD plus, well, it will catalyze an oxidation reduction reaction, producing pyruvate and NADH. Okay. So lactate dehydrogenase can be identified by this set of 4 numbers. So this first number 1 tells you that is an oxidoreductase. The second number tells you something about the groups that it reacts with. The third number tells you something about the substrates that it acts upon. And then this fourth number tells you specifically that lactate dehydrogenase is an enzyme that uses lactate and NAD plus, or it does the reverse reaction. Now I'm going to go back to this slide right here. So when you see 1.1, when you see, I'm going to hypothetically, if I were to say, hey, what type of, and the way that you would see this on an exam would be like 1.x.x.x is a EC number, enzyme classification number, for what type of enzyme. And from the information, and I'm showing you that on an exam, you would see only the first digit, 1.x.x.x. And I want you to say, oh, well, that's an oxidoreductase. Can you give me a Y or an N if that makes sense? Okay, great. Now, the next number, the second and third and fourth digits, I don't want you to try to commit those to memory, but what I do want you to know is what their significance is. If if the second digit is a 1, well, that is an oxidoreductase that acts on CHOH groups of donors. So if you see 1.1, it's an oxidoreductase because of this number, and then it acts on this CHOH group of a donor. If it's 1.1.1, well, it says it's an oxidoreductase that acts on a CHOH group and it uses NAD or NADP as an acceptor. And then this last digit right here, which is what we see here, this 0.27, tells you what is the specific combination of reactants and products. So give me a Y or N. Do you think that there is an enzyme that does what, or that, sorry, do you think that there is an enzyme that is 1.1.1.26? Y or N? Yeah, there's an enzyme 1.1.1.26. It uses, and sorry, let me rephrase that. So that enzyme does exist, but guess what? Or what could you say about this? Well, you could say that it's an oxidation reduction reaction, or it's an oxidoreductase enzyme. Next up is that it acts on a CHOH. It uses NAD or NADP. And the substrates here are going to be different. So it's an oxidation reduction reaction. I don't want you to know this number right here. I don't want you to know that complete number, but I do want you to know the first digit, the significance of that one. Could everyone give me an emoji in the chat if they understand that? Great. All right. So this enzyme in particular, this lactate dehydrogenase, this is an enzyme that, well, it helps after like when you're exercising or something like that. It's going to take the product of our pathway, pyruvate, our glycolysis pathway. It's going to take that pyruvate and make a bunch of this lactate, also known as lactic acid. Now, a transferase, well, what does a transferase do? That 2 indicates, when you see that, it is an enzyme that transfers a group. So, for example, it transfers a carbon, nitrogen, or phosphorus group. We will see phosphofructokinase, this enzyme, because it's a key important step in glycolysis. What this enzyme will do is it's going to take this substrate, fructose 6-phosphate, and ATP or adenosine triphosphate, and make fructose 1,6-bisphosphate and adenosine diphosphate, ADP. A hydrolase is going to use water to break a bond. That's all there is to that. And any hydrolase is going to do that. It's going to use water to break a bond. The end. Lyases will break bonds, carbon-carbon, carbon-sulfur, or carbon-nitrogen bonds, but without the help of water. Aldolase is an enzyme that we will see applying, or using that, or applying that. An isomerase, indicated by the number of 5, just takes all the atoms of a molecule and rearranges them, isomerizes them. Ligases, they form bonds between carbon and oxygen, carbon and sulfur, or carbon and nitrogen, and they combine that with the high energy bond breaking of a ATP molecule. So one important thing about a ligase versus a transferase. One thing that here's my kind of, okay, here is no, let's see. Okay. So a transferase, here's my transferase enzyme. And it's going to take a or molecule plus ATP. Transferase is going to take these two reactants and it's going to make this mol P plus ADP. Does everyone see how I transferred a phosphate from ATP onto mol P? If you don't follow that, if you don't see that, please say no. Okay. Now, the important takeaway is that did we break a bond on this molecule, this ATP? Yeah, we did. We took a phosphate group off of ATP and we moved that phosphate onto this substrate molecule. Now, if we look at a ligase, what we're going to do is we're going to have ATP, but the important part of this is that the phosphate, we're going to break this bond and this phosphate just floats off into the ether. It's not added onto our substrate like what we see here. Here's our substrate, and then here's our phosphorylated substrate, which is produced from a transferase. Okay, so what we're going to do when we look at all of the different steps of glycolysis, we're going to bring it back to these. Now, if, like I said, we're going to be, here's our learning objectives. I want you to identify examples of the 6 different classes of enzymes. I've given you those here, but we're also going to kind of inventory glycolysis and look at, okay, is this an oxidoreductase? Is this a transferase? Is this a hydrolase? So from here, what we're going to do, we've talked about the overall purpose of glycolysis. Basically glucose to pyruvate, and in the process, making ATP and NADH. Now, does anyone know, or could anyone add to the chat? If I got, let's say, pure potassium. Potassium is a group 1 element. If I got pure potassium and added it to a glass of water, what would happen? Tell me in the chat what would happen to that potassium added to that glass of water. And not a potassium ion, solid potassium. If I threw it in there, what's going to happen? And if you don't know, okay, perfect. It's going to combust. It's going to react very violently. The type of reaction that's going to take place is an oxidoreductase or oxidation reduction reaction. That reaction is going to be very violent, and it's just, I mean, that is kind of the violent reaction that's going to take place. Sorry. So when we talk about these reactions being oxidation reduction reactions in glycolysis, I want you to think about, oh, a release of a lot of energy. That's what's taking place here. So I think that that's a helpful way to think about, oh, okay, well, yeah, I'm ingesting sugar, I'm ingesting glucose, but what my body is going to do is basically try to get that stuff to explode. Okay, so in glycolysis, to put it simply, there's 2 phases. The first phase, we describe this as the investment phase. Okay. That phase begins with glucose. This molecule right here is glucose. Have we seen glucose before? Yes, we have. This is our cyclic form of glucose. We are going to invest a pair of ATP molecules. Now, I'm going to spend a bit of time on this slide going through each of the individual steps, and then I'll revisit all of these steps. I've got some poll questions, but I'm just going to talk about those individually. Today, I'm not going to be counting any sort of attendance or anything like that. So when we get glucose, we are going to have the enzyme hexokinase take a molecule of ATP and transfer a phosphate from ATP onto glucose to make this resulting molecule right here, which is also known as glucose 6-phosphate. So now what I want you to do is think about these classes. We've got one oxidoreductase, two transferase, three hydrolase, four lyase, five isomerase, or six ligase. Okay, we talked about the transferases, which I'm going to say two for our transferases and six for our ligases. Both of these required ATP. So I'm going to make a little table. I'm going to say needs ATP. I'm going to make this table. I'm going to say both enzymes 2 and 6, our transferases and our ligases require ATP. And then I'm going to say make ADP plus PI. Does a transferase produce ADP and PI? Well, let's take a step back. I can see right here that a ligase, enzyme class represented by the number 6, yes, it does. Okay, now I'm going to go back. Does a transferase? Well, a transferase makes ADP, but that other phosphate, the one ATP, that phosphate gets added to my substrate. So I'm going to say no, it does not make ADP and PI inorganic phosphate. Instead, a transferase is going to take that phosphate and put it onto something. So my question for you guys is hexokinase, is it a 2 transferase or a 6 a ligase? Add your response to the chat. Is it a 2 or is it a 6? Okay, I got one vote for 6, a ligase. I got a vote for 6 question mark. Well, let's think about this and let's look at our reactant on our product. We've got glucose and glucose 6-phosphate. Carbon number 6 on glucose has a hydroxyl group in glucose. Then hexokinase is going to take ATP and it's going to replace this hydroxide group with OPO3. So you could say that I'm transferring the phosphate from ATP onto glucose to make glucose 6-phosphate. Because my product here is ADP, not ADP plus PI. So now, since this is not the case, since I'm not making ADP and PI, that means that hexokinase is not a ligase. Hexokinase is a transferase because ATP, adenosine triphosphate, so that's a molecule with 3 phosphate groups. I'm taking one of those phosphates and adding it to glucose to make glucose 6-phosphate. Can you give me a Y if that makes sense or an N if that does not, and you'd like me to explain it again? So I'm going to say HK here, hexokinase, I'm going to erase these lines, is a transferase. Okay. Now, my next enzyme. And so when you're confronted with that sort of question, I think that a really good idea is to look at, okay, well, what are my products? What's being made here? And when you see, okay, here's my substrate, and here's one of my products, and that product has a phosphate group, that phosphate came from ATP, and that's what got us glucose 6-phosphate. Okay, my next enzyme is phosphoglucose isomerase. Take a moment and look at these different options. Do you think that phosphoglucose isomerase is an oxidoreductase, a transferase, a hydrolase, a lyase, an isomerase, or a ligase? How would you classify that based on its name alone? And then, if you're unsure by that, look at the two molecules. Your input is glucose 6 phosphate. Your output is fructose 6 phosphate. Okay, so does anyone know the difference between glucose versus fructose? Well, do they both have 6 carbons? Yes. One is an aldose and one is a ketose. So they're isomers of one another. So there's really no atomic difference between glucose and fructose. So then if we think glucose 6 phosphate, so that's glucose with a phosphate on carbon number 6, and fructose 6 phosphate, that's fructose with a carbon or with a phosphate on carbon number 6. Well, I think it's pretty reasonable to say that PGI is an isomerase. Okay, now what I'm going to do is I'm going to I'm going to give myself a blank slide to work with here. I'm going to ask you guys. Nope. Okay, so CH2OPO3 2 minus. I'm going to go down. Could you give me a Y in the chat if you agree that that is glucose 6 phosphate? I've got carbon 1, 2, 3, 4, 5, and 6. Okay, so there's my 6 carbons. Awesome. Now I'm going to rearrange this. I'm going to isomerize it. That isomerization is going to be completely reversible. So now what I'm going to do is I'm going to make let's see, a 5 carbon or a 5-membered ring. Okay, so I've got CH2, and I'm just going to move my OPO3, but for no reason beyond organization and spatial organization. I've got OH going down. So this is 6, 5, 4, 3. OH, that's my carbon 3, 2. Oh, OH, that's carbon number 2. And now this is carbon number 1, CH2OH. Okay. I'm going to erase my numbers 6, 5, 4, and 3. So this is just an isomerization. The next thing that I'm going to do is I'm going to be adding a phosphate. How am I going to add that phosphate? Well, what did I do for hexokinase? My first enzyme? I saw an OH group and I added a phosphate group there. I'm going to use ATP. And one of the products that I'm going to get is ADP. I'm going to get the same 5-membered ring. Organization, OH, OH, CH2, again, OPO3, 2 minus, then OH, and now I've got CH2, right, H2C, sorry, just organization. Okay. What type of enzyme do you think this is? Do you think it's a 2? So this step right here. Do you think that it is a 2 transferase or a 6 ligase? So since I used ATP and I took a phosphate off of that and I placed it right here on carbon number one, this is a transferase. My clue, or my biggest clue there, was the fact that my product was ADP and not ADP plus PI. Okay, so now what I have is I have this molecule right here. This is known as fructose 1,6 bisphosphate, or F16BP. That's what I've got right here. The enzyme that catalyzes this reaction is known as phosphofructokinase, or PFK. Sometimes you'll see it as PFK1. We're taking ADP and we're adding a phosphate group to fructose 6 phosphate to make F16BP or FBP. The next thing that we're going to do is we're going to break that molecule in two. When you look at this molecule right here, I'm going to erase everything except for the FBP. That fructose 1,6 bisphosphate molecule that you see there, I want you to look at it and say, ah, that's kind of symmetrical. Because what we've got is, well, something that we can kind of split in two. When we split it in two, we end up with these two molecules. Glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, or DHAP. GAP and DHAP. Do those molecules look kind of similar to one another? Well, they should. One of them, or they are both 3-carbon molecules. One of them is an aldose, one of them is a ketose. Glyceraldehyde 3-phosphate, this is an aldose. And dihydroxyacetone phosphate, this is a ketose. So we've got an aldose and we've got a ketose there that result from our fructose 1,6 bisphosphate splitting. Now, this poll question brings back that terminology, aldoses or ketoses. Okay, so we have what term is best used to describe the sugar below. Glucose is our molecule. Glucose is an aldose. It's an aldehyde. It's got an aldehyde functional group. Is it a ketose? No. Is it a trodose? No. Is it a pseudose? No, it's not. Okay, so like I said, the first phase of glycolysis is the investment phase. We invested a pair of ATP. Now, the next phase of glycolysis is sometimes referred to as the payoff or the return phase. We're going to begin that phase with glyceraldehyde 3-phosphate. I'll get more into why that is our starting point in just a little while, but we're going to invest 2 NAD plus, 2 PI. We're going to get back a pair of NADH, and we're going to get back a grand total of 2 ATP and then 2 ATP. Is that a typo? Oh, no, it's not a typo. Instead, we are getting a grand total of 4 ATP back. So in the second phase of glycolysis, you get 4 ATP back. You started by investing 2, you get 4 back, which brings you to your net ATP return of 2. Think about it kind of like a business. So in summary, our reactants are glucose, inorganic phosphate, NAD plus, and ADP. Our products, this is the net products, are 2 pyruvate, 2 NAD, or sorry, 2 ATP and 2 NADH. The most important product are those 3. I didn't talk about the proton and the water because they're a little bit less important. So when you talk about glycolysis, what I want you to do is think about, well, I'm investing ATP, I'm getting back pyruvate, ATP, and NADH. So in summary, this is all of it. There are a total of 10 enzymes. We're going to go one by one through each one of the enzymes. That link that I shared a little bit ago shows all of those enzymes broken down into individual YouTube shorts, each step. Here it is, simplified as much as I possibly could. Is there more information than what I could put in 31 seconds? Yeah, but I wanted to hit on the high points of what each step was. So here we go. Hexokinase, the first enzyme in glycolysis. This is a transferase. We are transferring a phosphate group from ATP onto glucose to make glucose 6-phosphate. The name is hexokinase. Although this shows an equilibrium arrow going backwards, this is considered to be an irreversible reaction. So I'm going to say IRR, irreversible. Does it require magnesium? It does. Is that something that I want you to drill into your memory? Not so much. What I want you to focus on is that it is a transferase. It transfers a phosphate from ATP onto glucose, resulting in ADP and a proton. So ATP plus glucose yields glucose 6-phosphate and ADP. That's what hexokinase does. Next up is our isomerization. We're taking our aldose, our phosphorylated, and I'm just trying to use some interchangeable terms here. Our phosphorylated aldose, glucose 6-phosphate, and we're going to produce a phosphorylated ketose, known as phosphoglucose isomerase. This is just rearranging. This is an isomerase. Next up is our second phosphorylation. So our second ATP that we're investing. This is a transferase. It's going to take ATP. This is also an irreversible step. We're going to take a phosphate off of ATP and put it onto fructose 6-phosphate to make fructose 1,6-bisphosphate, or FBP. Now, we have a doubly phosphorylated molecule. What we need to do with that molecule, this is doubly phosphorylated, it's almost symmetrical. We're going to split it into two sugars, or two 3-carbon molecules, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. The enzyme that does this is an aldolase. Look at that EC numbering. Okay, EC numbering of 4. Oh, well, all that we care about is the 4. What type of enzyme does that? Well, we are breaking a carbon-carbon bond. So this is a lyase. So I'm going to go back and I'm going to circle the carbon-carbon bond. So this is a lyase. What bond is being broken? Well, the bond between carbons number 3 and carbon number 4. We're splitting that. And so the result is these top 3 carbons, 1, 2, and 3, become these right here. This carbon number 1 right here is this carbon right here. Now, if you take metabolism, you'll talk about carbon numbering and labeling with radioactive isotopes. We're not going to be talking about that in this class. The bottom half of this molecule, which I'm going to put a square or a rectangle around, well, this phosphorylated carbon right here is this one right here. So we get an aldose and a ketose. Does everyone, could you give me a Y or an N in the chat if you see that this, can you see the aldehyde functional group? Can you see the ketone functional group? A Y or N to the chat. Great. Okay. Now, the important takeaway is that we're splitting a 6-carbon molecule into 3-carbon or 2 3-carbon molecules. That's an important point because at the end of all of this, what are we going to end up with? 2 3-carbon pyruvates. Now, the best term to describe this sugar below, okay, we've got carbon, carbonyl, carbon. This is a ketose. This carbon number 1 right here is an aldo or aldehyde, so this is an aldose. Now, what we want to do for the second phase of glycolysis right here is we want to make and start with a pair of glyceraldehyde 3-phosphate molecules, GAP molecules. So what that means is, sure, we broke this into 2 molecules, a glyceraldehyde 3-phosphate and a dihydroxyacetone phosphate. But what we need to do is we need to convert dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. So when we use this lyase to split then we're going to take one of them and convert it to the other so that we are leading with, or we're going forward with two copies of glyceraldehyde 3-phosphate. So that's what this is showing. With the dihydroxyacetone phosphate that's produced, we have to isomerize it to make glyceraldehyde 3-phosphate. And the enzyme that does this is triose phosphate isomerase. And if you're playing along at home, you guessed it, this is an isomerase. So once we've used this reaction, we are done with the investment phase. So enzyme number 5, triose phosphate isomerase, marks the end of the investment phase. So if you're thinking about this in terms of classes of enzymes, we have a transferase, we have an isomerase, we have a transferase, we have a aldolase, which that's breaking a carbon-carbon bond. This is a lyase. So transferase, transferase, lyase, and then we're going back to an isomerase. So those are just the classes. Now, if you're thinking, well, what's more valuable for me to know? The names of the enzymes? Yes, the names of the enzymes, but I think that it's helpful to kind of remember what's taking place so that you can understand those, the functions of those different classes. Okay, so as soon as we've got our two molecules of glyceraldehyde 3-phosphate, which you'll see it written, you might see GAP, G3P, GA3P. Those are all kind of interchangeable. What are we going to do? We are going to take an inorganic phosphate molecule, a PI, and we are going to add it to our glyceraldehyde 3-phosphate molecule. How are we going to do that? Well, we're going to use NAD+. This is an oxidoreductase. A good clue for an oxidoreductase is whenever you see the name dehydrogenase, I want you to think oxidoreductase. I want you to, whenever you see NAD+, I want you to also think oxidoreductase. So we are adding a phosphate, but that doesn't mean it's a transferase because this inorganic phosphate, this PI, is not coming from anywhere. It's just sort of floating out in the ether. It's not coming from a donor molecule. It's just present. So we are making NADH. We are also making this molecule right here, 1,3-bisphosphoglycolate. So this molecule right here is 3 carbons, and it has 2 phosphates. Now, my question for you guys is, how many of these molecules from the original glucose that we started with, how many of these do we have? We started with 1 glucose. How many of these right here do we have? Add your number to the chat. I'll give you a hint. It's not 3 or larger, and it's not 0. So we started with 6 carbons of glucose. We split that into a pair of 3 carbon molecules. So we had 2 glyceraldehyde 3-phosphate molecules. So this inorganic phosphate, we have 2 of them. So this NAD+, we've got, you guessed it, 2 of them. How many NADH are we producing? 2 of them. At this point, when we talk about the return phase, every single reaction that you see, it's happening in duplicate. So how many 1,3-bisphosphoglycolate molecules do you have? 2. So that 1,3-bisphosphoglycolate, what we are going to do is we're going to use this enzyme right here, phosphoglycerate kinase, and ADP. And we're going to take this phosphate that we just added, but phosphoglycerate kinase is going to take that phosphate off and add it to, or transfer it from 1,3-bisphosphoglycolate onto ADP to make ATP. So how many ATP do we have? Two. How many 3-phosphoglycerates do we have? Two. This is a transferase. We are transferring the phosphate from 1,3-bisphosphoglycolate onto 3-phosphoglycerate. And we've got two of them. So if you're thinking about the accounting, tell me in the chat, and I need a response here. Tell me in the chat, how many ATP did we invest in our investment phase? We invested 2 ATP. Perfect. How many have we gotten back so far? Add it to the chat. 2. So if we were looking at this pathway as a business, we've broken even in terms of our ATP investment. So that's great. In addition to that, we've got 2 NADH molecules, which will be valuable down the line. But at this point in time, after phosphoglycerate kinase has catalyzed its reaction, we've broken even. We've got 3-phosphoglycerate, which we are going to proceed with. We've still got a phosphate floating around on this molecule. That 3-phosphoglycerate, we're going to rearrange it to make 2-phosphoglycerate, or 2PG. This is an isomerase. We're just moving the phosphate from carbon 3 onto carbon number 2. Okay. Now, that phosphoglycerate mutase that does that reaction primes everything for this 2-phosphoglycerate to then be acted on by this enzyme enolase. Enolase, okay. Well, what are we doing? We're making water. We're releasing a water. The enzyme is phosphoenol pyruvate. Now, that phosphoenol pyruvate that we're generating is an important high energy molecule. Whenever we are generating this water, this enolase is a lyase. The important thing here is that this is not a hydrolase because we're not breaking a bond by adding water. Instead, we're releasing water. So this enolase is a lyase. Our last step is phosphoenol pyruvate being acted on by this enzyme pyruvate kinase. What we are doing is we're transferring the phosphate from phosphoenol pyruvate onto ADP, making ATP. Tell me in the chat how many ATP. So how many ATP have we made with this last step? We made a pair. We made 2 ATP. How many pyruvate? 2. So our output is, well, phosphoglycerate kinase got us 2 ATP, and that got us back to, break-even money. Pyruvate kinase generated 2 more ATP. So when it's all said and done, we have a net of 2 ATP because this step, this transferase right here, got us to 0, and pyruvate kinase got us to profit. So whenever you talk about glycolysis and ATP production, people will talk about total ATP production and net ATP production. The total ATP production from glycolysis. Could you tell me in the chat what is our total ATP production from glycolysis? 4, perfect. And then the net is 2 because we had to invest 2. All right. Now, this is glycolysis as a whole. There are 3 steps that make this pathway an overall kind of irreversible pathway. Those three steps are right here. They are hexokinase. I'm going to scribble that out because that's effectively zero. Hexokinase is an irreversible step. Phosphofructokinase is an irreversible step. And pyruvate kinase is an irreversible step. So the two steps where you are generating or you are using ATP are irreversible. One of the two steps where you are generating ATP is irreversible. There are a total of three enzymes known as kinases. Now, all of these kinases are indeed known as transferases, but only three of them are irreversible. Phosphoglycerate kinase is a completely reversible reaction. Phosphoglycerate kinase is where we take 1,3-bisphosphoglycerate to make 3-phosphoglycerate, or 1,3-bisphosphoglycolate to make to make 3-phosphoglycerate. Okay, now where is this taking place? Where is glycolysis taking place? It's taking place in basically every cell in your body. Now, does anyone know the organ that is most responsible, or does anyone know of an important organ for regulating your blood sugar? And add it to the chat. I'll give you a hint. The one that I'm thinking of starts with an L and ends with an iver. Liver, perfect. Your liver is very important for regulating your blood glucose level. Your blood glucose levels typically, in terms of concentration, it's about 5 millimolar. Now, in most cells, you have glycolysis taking place, and the enzyme that's driving it, your first enzyme, is hexokinase. Muscle cells, they have hexokinase. If you give them fructose, they are going to be able to metabolize that 6-carbon molecule, because if you look at that name, hex, meaning six, hexose, 6 carbons. If you give your typical cells fructose, mannose, galactose, they're going to be able to do something with it. And they have a Km, or hexokinase has a Km of 0.1 millimolar. So it doesn't take a lot of sugar for hexokinase to be activated. Within your liver, which more closely monitors your blood sugar level, you have an enzyme known as glucokinase. Now, glucokinase is very selective. What it will do is only act on glucose. So if you were to give glucokinase, if you were to purify glucokinase and feed it a bunch of fructose or mannose, it's not really going to act on it with any high capacity or with any sort of activity. It has a Km of approximately 10 millimolar. Okay, so could anyone tell me glucokinase, if you were to give glucokinase 10 millimolar glucose, could anyone tell me how fast glucokinase is going to be catalyzing its reaction? I'm going to label glucokinase as GK. How fast is GK? I'm just going to say, how fast is GK going? Well, the answer is, if you give it 10 millimolar, it's going to be going at Vmax divided by 2. If you agree with that, give me a Y in the chat. If you don't understand that, give me an N in the chat. Okay. So if you give it 10 millimolar glucose, then it's going to be going this fast. Got it. If you, however, give it, so I always like to think about a Km as like a payment. If you're paying glucokinase 10 millimolar glucose, it says, okay, cool, I can go half as fast as my as I can. half as fast as possible. If you then approach glucokinase and say, hey, I got 2 millimolar glucose, first of all, is 2 smaller at an S, or is it L larger than 10? Just the numbers. Yeah, it's smaller. Okay. So you already know glucokinase needs 10 millimolar. If you give it 2 millimolar, is it going to go faster or slower than if you gave it 10 millimolar? Give an F for faster and S for slower. It's going to go slower. It's going to say, I need 10 millimolar and you're offering me 5 millimolar? Get a life, man. No, thank you. So my point there is glucokinase. If your blood sugar is maintained at about 5 millimolar, well then glucokinase is like, okay, cool. I'll do the reaction, but I don't need to do a lot of it because the blood sugar level is, if it's maintained at 5 millimolar, there's not much reason for me to do anything. I don't want to do anything, so I'm not going to need to do anything. Could you give me a Y in the chat if that makes sense, or an N in the chat if that does not make sense? Okay. Now, hexokinase and glucokinase are two enzymes that do the same thing. The difference is why is one, or like where are they located, and why is one going to be activated than the other? If you go and get, let's say that you get a bunch of Coca-Cola and a bunch of, let's see, yeah, Coca-Cola and Skittles and then M&Ms. Well, M&Ms have some fat from the chocolate. So let's just say that you get a bunch of Skittles and a bunch of Coca-Cola. Then your blood sugar might go up. Then glucokinase might be like, hey, the blood sugar has gone up from our normal 5 millimolar to 7.5 millimolar. I'm not going at half my maximum speed, but I'm a little bit more aware of what's going on. So I'm going to be reacting a little bit more. If you continue eating a bunch of sugar and eating a bunch of Skittles, well, then glucokinase might be like, whoa, this blood sugar level still seems high and is sustained at a high level. I need to remain active. Okay, now I want to go back to our learning objectives and just kind of inventory these things. So, summarize the net reaction of glycolysis, including the reactants, products, and energy yield. And outline our 2 phases. Okay, so our 2 phases of glycolysis, the investment and return phase. Summarizing our net reaction of glycolysis, I want you to know the reactants and products, and our energy is yielded in the form of our ATP and NADH. I got one more slide after all of this that I want to go over the NADH. 10 key enzymes. We've talked about those. Recognize the 3 irreversible steps. That's HK, PFK1, and PK, or pyruvate kinase. Yep, those are all here. Then describe the hexokinase, glucokinase, and glucose phosphorylation and how their activity differs. Well, it has to do with the tissues and their sensitivity. Calculate the ATP yield of glycolysis investment, okay, in aerobic and anaerobic conditions. Now, and then explain the role of NAD plus in glycolysis, how it's regenerated in aerobic and anaerobic conditions. Okay, so first of all, when we look at this pathway, this entire pathway right here, what I want you to notice is there's no O2 integrated in any one of these steps. So this is an O2 independent process. This is an anaerobic process. What we will do is move on after all of this, and oh, dang it. We will take on this step right here, or this process right here, the electron transport chain. The electron transport chain, if you look at this, this is a process that takes place in the mitochondria. This is where that term of the powerhouse of the cell really comes from. The NADH that we generated in our glycolysis and that will be generated later on in the Krebs cycle or the tricarboxylic acid cycle is going to be used to transfer a bunch of electrons. Now, is the transferring of electrons, is that an exciting process? Could that yield a lot of energy release? Add a Y or an N to the chat if it yields a lot of energy, an N if it does not yield much energy and is kind of boring. When we transfer electrons, yeah, there's a lot of energy that's released. So this NADH has a lot of energy to release. It's going to release some electrons. It's going to transfer these electrons. They're going to go through these different protein complexes until ultimately we generate a bunch of ATP. This is an O2 dependent process. So this electron transport chain, that's where you really make all of the money or you make all of the ATP from the breakdown of a sugar like glucose by producing NADH. Sure, in glycolysis, you're like, hey, I made some ATP. Exciting. That's awesome. But NADH is really the heavy hitter when it comes to generating a lot of energy. Okay, we will get more into this a little bit later on, but for the time being, that's what I've got for today. I'm going to go ahead and stop.